Oh, we lost Jorge again. Okay.
Listo, estamos en vivo.
So we're live now. We're going to start. I'm going to give a few minutes before starting the talk. Vamos a esperar unos.
Yes. Let me know when I should share it.
Sure. Vamos a esperar unos minutos antes de comenzar la presentación. Gracias. Jorge? Are you there, Jorge? Can you hear us, Jorge? You have the microphone silenced. ¿Nos escuchas, Jorge? Jorge, el micrófono está apagado. Ah, ok. Pueden, Cristina, Cristi, creo que está desactivado, Jorge, creo que no puede habilitar su micrófono.
Sí, ya le mandé la solicitud desde hace un momento, pero no me la acepta.
Ah, ok. ¿Te llegó la solicitud, Jorge?
Sí, ya le mandé.
¿Dónde está? Pues me imagino, a ver, pon el participantes o el chat, alguno de los dos, a ver si ahí aparece. Yo imaginaría que te debería aparecer en la pantalla principal.
Ah, creo que ya. Ya.
So, whenever you're ready, Jorge, we can start.
Yeah, 1 second. Wow. Okay, just Eduardo, y si pierdo ¿cómo se llama? La conexión, por favor te voy a encargar las preguntas, porque esto está bastante mal.
Sí, claro que sí.
All right. Oh. Okay, let's start today's colloquium. Thank you very much, Raimund, to be with us today. Raimund Dutzler has been a full professor of biochemistry at the University of Zurich, Department of Biochemistry, since September 2009. Raimund Dutzler studied biochemistry at the University of Vienna and received his PhD in biophysics from the Biocentrum of the University of Basel. He conducted his postdoctoral training at Rockefeller University under the direction of the Nobel Prize winner Roderick MacKinnon. He studies the mechanism of transmembrane ion and lipid transport by combining biochemistry, X-ray crystallography, cryoelectro microscopy, and electrophysiology. Dr. Dutzler has elucidated the structure of different ion channel and transporters and has made seminal contributions to understanding the mechanism of ion and lipid transport. Today, Dr. Dutzler's seminar is entitled The M16 Family of Calcium-Activated Lipid Scramblases and Ion Channels. I will ask the audience to please write down your question, and me or Eduardo will read it to Dr. Dutzler so he can answer the question. Dr. Dutzler, thank you very much for accepting our invitation. Welcome to San Luis Potosí virtually, but hopefully we'll be okay. Thank you very much. Go ahead.
Yeah, Jorge, I hope you understand me.
Yes,
I will start to share my screen now.
Mm-hmm. Yep.
So. Yeah, I think before we start, thanks for inviting me. I think it's wonderful that we can communicate on such a large distance. Of course, it would be nice to be there, but getting there is difficult. So I think this is a good compromise. The last time when I have been in Mexico was at 2017. There was a thematic meeting on ion channels by the Biophysical Society in Mexico City. It was my first time there, and I really enjoyed it enormously. The meeting was intellectually stimulating. The city is beautiful, and I enjoyed the museums. And you know, I was impressed by the level of ion channel research in Mexico. So I would like now to tell you about, you know, and give you an overview of 13 years of work on a protein family. And the protein family, as Jorge has mentioned, is called the M16 family. So the M16 is, you know, the name is for transmembrane protein of unknown function number 16. And this kind of a bit unusual nomenclature originated when, after the sequencing of the human genomes, membrane proteins were assigned by hydropathy analysis. But many of them really could not be assigned to a function because they were not related to anything. And so they were given this M16 nomenclature. So the M16 proteins are interesting and unusual because they catalyze two functions which at first glance do not look as if they would be related. And the first function is ion conduction. We are talking about anion conduction. And this is a process which has been described for decades and been studied for a decade. This is an ion channel which is activated by an increase of the intracellular calcium concentration. And this protein was known to be involved in chloride secretion in epithelia, for example, in the epithelia of the airway. And there, the calcium-activated chloride channel is in the same epithelium as the CFTR channel. So for this reason, it's thought that if you would kind of activate this protein, eventually you would be able to compensate for the loss of chloride conduction in cystic fibrosis. But other places where calcium-activated chloride channels are important are, for example, in endothelial smooth muscles, where the calcium-activated chloride channels are involved in electrical signaling. They also are supposed to play a role in olfaction and eventually also in pain reception. The other process is related to this process of chloride permeation only by the fact that the protein is activated by calcium on the intercellular side. And we are talking here about lipid scrambling. So what is lipid scrambling? A lipid scrambling is a process which is catalyzed by a scramblase. And the scramblase is something like a channel for lipids. So the lipid composition of the plasma membrane is asymmetric. So we have different lipids on the inner leaflet compared to the outer leaflet. And this asymmetry is established by an ATP dependent transport process. So we have ATPases, which transports negatively charged lipids like phosphatidylserine to the inside. And, you know, they remain on the inside unless there is a process where the asymmetry is dissipated. And this is happening by a scramblase. And this process is important because during this process, then the negatively charged phosphatidylserine gets exposed to the outside and it's recognized. And this is, for example, happening during the initiation of blood coagulation, which is initiated by a calcium-activated lipid scramblings. Another process would be apoptosis, but there's a different family of channels of scramblases which is involved in apoptotic scrambling. Other processes where calcium-activated scramblases are activated is, for example, in membrane fusion of myocytes, of trophoblasts, or of osteoclasts, or in exocytosis, the release of exocytotic vesicles. So at first glance, it doesn't look like these two processes would necessarily be catalyzed by the same protein family. But if you have a closer look, then it's clear that in both cases, we are facing an electrostatic problem. Because we want to, you know, transfer hydrophilic groups across the low dielectric of the membrane. You know, whether it's now an ion or it's a lipid head group. In both cases, we face a large energetic obstacle because we have to kind of strip the water, which is tightly interacting with both moieties. And so we have a large energy barrier, and the large energy barrier usually kind of slows down the kinetics. So what we need is then a catalyst, an enzyme, which facilitates this, which in case of ion conduction is a process we are kind of very familiar with. It's an ion channel, which then allows an aqueous pathway to compensate for the interaction with water. In case of a scramblase, it was not so clear how this might happen, but there has been a proposal by Anand Menon's group. They have suggested that this might happen by a so-called credit card mechanism. And so he has postulated that the scramblase would offer kind of like a half channel, which is exposed to the membrane, and which would accommodate then the lipid head group and provide a pathway for the lipid head group, because the apolar chains might reside in the membrane. So both processes, calcium-activated chloride conduction, calcium-activated lipid scramblings, were known and were studied for a long time. But the molecular identity of the protein which catalyzes these processes has been unknown for a long time. And it's only in 2008 that 3 groups independently, the group of Lilian, the group of Utek O, and the group of Louis Gallietta, discovered the protein TMEM16A as the calcium-activated chloride channel. And then by just homology, it became clear that this is a protein family which is only expressed in eukaryotes, but we have 10 close paralogs in humans. And they are named TMEM16A to K. And initially, it was obvious that, you know, due to the high sequence relationship, it was kind of thought that it's clear that all of these are calcium-activated chloride channels. But only 2 years later, the group of Shigegatsu Nagata in Japan identified TMEM16F as the protein which is responsible for the calcium-activated scrambling of lipids during blood coagulation. And this was initially, of course, controversial because, you know, how can it be that such closely related proteins can do such similar things? And it was then only in 2013 that Alessio Accardi's lab purified a fungal homolog and reconstituted and could show then on the isolated reconstituted system in vitro that this protein indeed scrambles lipids. And now we know that only 2 of these family members, the proteins TMEM16A and TMEM16B are chloride channels, and the others are lipid scramblases. But among the lipid scramblases, we have 2 different clades, ones which are closer related to the fungal homologs. They are residing probably in intracellular vesicles, and another clade which are more related to the protein TMEM16F, the calcium activated scramblase in blood platelets, which initiates blood coagulation, and they are more related to the channels TMEM16F. So, and it was about that time that we also kind of, when TMEM16A was discovered, that we became very interested, because, we had historically a strong interest in chloride channels, chloride transport, and in a way we were waiting for this protein to be discovered. But this was the time before cryoelectro microscopy. So the only way of determining structure was to use X-ray crystallography, which was extremely tedious. And so the only kind of systems which were accessible to us were the fungal proteins. And for that reason, we screened many, many different fungal proteins. And we found then one of the 100 proteins which we screened, which was from a fungus called Nectria hematococcus, or NH-TMEM16, to behave biochemically reasonably well, so we could proceed with characterization. And NH-TMEM16 is working as a scramblase, and I'm showing you just the assay to show this. This is an assay which was initially probably developed by Anat Menon's group. So what we are doing is we reconstitute the protein in liposomes, and then these liposomes contain fluorescent lipids. And here we would have a liposome without any protein, and we have the fluorescent lipids on the inside and the outside. Now, if we put a reducing agent on the outside, we take the membrane impermeable dithionate, then we would bleach all the fluorescent lipids on the outside. And what we get is here a reduction of the initial fluorescence to 50% here over time. If, however, we have a scramblase like NH-TMEM16, then this scramblase would catalyze the movement of lipids on the inner leaflet to the outer leaflet, and we get a further reduction here, as is done for NH-TMEM16 in the presence of high calcium. Now, this process is calcium dependent, because in the absence of calcium, we see a much reduced kinetics of this scrambling, and this kinetic is then increasing in a calcium dependent manner. So you see that even in the absence of calcium, the protein appears to be active. And this is not an artifact. It's really that probably these intracellular scramblases, these fungal scramblases, are not so tightly regulated by calcium. We then proceeded and determined the structure of NH-TMEM16 by X-ray crystallography. This was, a heroic effort. It took us about 4 years. I think we prepared over 2,000 liters of yeast cell culture to make crystals and were able to determine the structure. And this is what we found. We found a dimeric protein. You see there are 2 subunits. Each subunit has 10 transmembrane spanning helices. And the region which is interesting is here really at the periphery of each subunit. So we have 2 catalytic regions which are responsible for the scrambling of lipids. And we have another thing which is very important. This is this indicated by these blue spheres here. This is the regulatory calcium binding site, and the regulatory calcium binding site is actually located within the transmembrane domain inside the membrane. This is the case for all the family. So the fact that we have kind of 2 subunits which are probably functionally independent, we can focus on one subunit, and we are looking here at kind of the transmembrane domain from the outside. I mentioned we have 10 transmembrane helices, but the catalytic unit is only kind of encoded by helix 3 to 8. And we are looking here from the outside, and here we are looking from the direction from the membrane in this direction. And you see I have colored 2 helices here in red and in blue. And this one is helix 4 and the other one is helix 6. And I just want you to kind of remember these, because these are 2 kind of very important regulatory elements in many of the different proteins I would like to talk about today. And in the case of NHT-MEM16, both helices form the edge of a membrane-accessible groove. And this membrane-accessible groove we call the subunit cavity. So the subunit cavity is a polar cavity which spans the entire membrane from the outside to the inside. Its surface is polar, which is unusual for the surface of a membrane protein, and it's shown here, all the residues shown in color here are polar or charged residues, and it has about the size of a lipid head group. The other feature which is close to this polar cavity is the regulatory calcium binding site. So if we kind of look at the cavity, rotate by 90 degrees, you see this would be the membrane boundary. This calcium binding site is located in the inner leaflet of the membrane. And we were able to identify this calcium binding site by crystallography. And you see here there's a special experiment, it's called anomalous scattering, where we can really kind of identify these calciums without any doubt. You see that we have 2 calcium ions bound, and they are coordinated by 3 alpha helices, and they contain here, these helices contain negatively charged residues which are highly conserved in the entire family. And here there's also a non-charged residue. And you see that one of these residue is this blue, one of these residues here is kind of donated by this blue colored helix 6. And this is the important helix, you know, it's the one helix which I have shown you at one of the boundaries. And this is a very important gating element. So what we think we know now from the structure is a way how kind of, you know, lipids probably, how the protein facilitates the diffusion of lipids across the membrane. I just show you here a very simple path calculation where you just should see that, you know, this is about the dimension of a lipid, a head group, and you see it fits well in the cavity. So what we have never seen here is kind of a defined ion binding site. So it doesn't look like that you would have, you know, ion binding site, not ion, lipid binding sites here in this cavity. It looks really like a delocalized polar pathway. We were then very interested in the question is, how does calcium regulate the activity of this protein? And this was very difficult to study by X-ray crystallography because the protein was not very stable in the absence of calcium. So we had to turn to cryoelectro microscopy and study then the protein in lipid nanodisk. And this means that you study it in a membrane environment, which is particularly interesting for scramblers because the lipids are also the substrate. And what we have found is that, you know, in NHT-MEM16, in the absence of calcium, we found that the subunit cavity has collapsed. So the 2 helices, alpha helix 4, alpha helix 6, which were distant in the active conformation, in the calcium-free conformation have collapsed. And if we kind of studied the protein in a calcium-bound conformation, then we actually found an ensemble of 3 conformations, one in a closed conformation, one in an intermediate conformation, and one in an open conformation, which is basically identical as we have seen it in the X-ray structure. And what you see here is a morph between these conformations, just to give you an impression of how this activation process is working. So we are going from a calcium-free conformation, calcium binding, a kind of, you know, tightening of alpha helix 6. And this is then coupling from alpha helix 6, where the calcium binding site is, to alpha helix 4 to open the cavity. And this is just a surface representation, which just shows you the very same thing. So we go from the closed, you know, conformation where the 2 helices are interacting to the open conformation where they have separated. So apart from opening this cavity, there's another feature which also is probably providing an essential feature for a lipid scrambling. And this is that the protein distorts the membrane. And this is visible here, kind of, you know, we are looking here at low pass filtered maps of the protein in lipid nanodiscs. And you see the surrounding lipids, which are here colored in kind of green and yellow. They are not straight and even, they are bent. And, you know, we are slabbing in, then you see that, you know, this would be the 2 head group regions of the lipids. You see that we get a thinning and a depression of the lipid towards the side of the subunit cavity. And here we see it also on the other side, on the intracellular side, on the extracellular side. And this means that the protein distorts the surrounding lipids, and it steers the lipid head groups into the subunit cavity. So it kind of, by this distortion, it lowers the barrier for lipid head groups to cross the membrane. And you see here this density of probably lipid head groups delocalized in the subunit cavity. And this was for us an amazing thing to see, you know, and Alessio Arcadi found very similar results than for a related protein. Okay, so this means that our studies on NHT-MEM16 gave us a first impression of how a lipid scramblase might work. And the next big question we were interested, how is this related to how the channel works, T-MEM16A? And you know, for T-MEM16A, this doesn't exist in yeast. So the channels only exist in higher eukaryotes, so we had to express them in hex cells, and you know, we wouldn't have been able to proceed here with crystallography, so we really had to study this with cryoelectro microscopy. And this is what you see here. This is the structure of T-MEM16A in a calcium-bound form determined by cryoelectro microscopy. You see, it's also a dimer, so it's the main features are actually pretty similar. Of course, there are kind of, you know, details which are a bit different. So we have more region here on the extracellular side, but you know, also we have the dimer, we have the ion binding sites here. The real difference between NHT-MEM16 and T-MEM16A is in the subunit cavity. And what you see here is not the conformational change of a protein, it's a morph between the helix arrangement in NHT-MEM16 and T-MEM16A. And you see that in NHT-MEM16, the cavity is open, and in T-MEM16A, you know, alpha helix 4 has rearranged to engage with alpha helix 6 to form a protein enclosed aqueous pore. Mm-hmm. And this is what we understand now about, you know, ion conduction in T-MEM16A. This is kind of by work of many excellent groups. Jorge has contributed this in very kind of beautiful ways in seeing how the protein functions. We have done quite some experiments. And I just gonna guide you through and just explain you what we think how this protein works. So you see that we have two ion conduction pathways, each one on each side, you know, enclosed in each subunit. So and we have done some experiment, and Sang-Yu Chen has also done similar experiment to show that these pores are essentially independent. So there's not much crosstalk between these two pores. So at the end, we can focus on one pore to see what's going on here. So this would be, you know, this would be the rough structure of the pore lining. We have a pore which has an hourglass-like shape. So we have larger vestibules on the extracellular side, on the intracellular side. So they are large, they are aqueous. They actually have an excess of positive charged residues. So they're very hydrophilic. But then the two vestibules are leading into a narrow region. And we call this the neck region. And this neck region is amphiphilic. So it contains hydrophobic residues, as you see here, and no charged residues, but predominantly polar residues. And we think that both parts of the pore contribute to the selectivity for ions. So the positive residues here to attract the anions, and maybe this narrow part to discriminate between anions and cations, because, you know, anions are usually larger, so the energy of dehydration is usually, you know, less costly than for cations. And kind of the structure of this pore kind of, you know, reflects several properties what's known about the channel. So we know the channel is highly selective for anions over cations. So we basically don't get cations conducting, but it does not discriminate well between anions. So you have anions like isothiocyanate, probably also bicarbonate, which would permeate. The second thing is we don't really find an ion binding site here. So there's not the site in this narrow neck region where we bind an ion. And this is reflected in the fact that if you just kind of look at the dependence of the conduction on the conductance on the ion concentration, that you know, if you increase the salt concentration, you get a nearly linear increase in the conduction. So there's very little saturation. And then what we also know is that the channel is not very kind of, the single channel conductance is not very high. So it has only about 2.5 picosiemens as estimated by non-stationary noise analysis. So, and this is kind of going hand in hand with kind of probably that we really don't have a very wide channel. Nevertheless, if you map the pore diameter here, then you will see, and if you compare it to the radius of a chloride, then you will see that at some regions, you know, this channel is more narrow than you would expect it to be for a chloride. So we don't think that we have a fully activated channel, but we think that, you know, even, you know, and we think that the channel has to expand to activate, but we don't think that it gets much larger, so that it remains a narrow channel in the center at least. The calcium binding site of TMEM16A is kind of very similar, you know, the same position as in NH-TMEM16. We have again the free helices, alpha helix 7, 8, and 6, the gating helix. You know, we have 2 calcium ion bonds, we have conserved negative charges. So the location of the calcium binding site in the inner leaflet of the membrane means that the calcium binding site is within the electric field of the membrane. And this means that the calcium, you know, either gains energy or has to spend energy to reach its binding site depending on the membrane potential. And as a consequence, the activation of the protein by calcium is voltage dependent. You see here, this is a calcium concentration response relationship from excised patches. And we see that at 80 millivolt, we activate the channel with, you know, sub-micromolar potency. And this process is cooperative, so we have a Hill coefficient of about 2. And this is because probably we have 2 calcium which are binding. At negative voltage, you know, calcium has to spend some energy to reach its binding site, so we need a higher concentration to saturate the conduction. But, you know, in both cases, so, and this means that if you are looking at an intermediate concentration, then, and this has been observed for a long time, then it looks like as if the open probability is voltage dependent. And this is because probably you have then less calcium bound at negative than at positive voltage. But at kind of, you know, at saturating calcium, the current voltage relationships are more or less linear. Okay, we were then interested in the question is, how does calcium activate the channel? And so we determined the structure of TMEM16A in the absence of calcium. And what we found is, you know, here we are looking at the pore region, again, from kind of a view on the subunit cavity. Here we have alpha helix 4 and alpha helix 6. We see that, you know, we did not see much changes here, but we saw a pronounced change in alpha helix 6. And this makes sense because in the calcium bound form, alpha helix 6 contributes to the calcium binding site. You see here this negatively charged residues interacting with both calcium in the absence of calcium. But we think that this kind of locks alpha helix 6 in a strained conformation. And if you then kind of wash out calcium, then you have first the repulsion of all these negatively charged residues. And additionally, we know that this alpha helix 6 has kind of an amino acid which is in addition to the scramblase. So in order to bring this residue in interaction, you have to unwind the helix. So you form a pi helix. And this puts a strain. And when you don't have the calcium, then the strain is released and kind of, you know, alpha helix 6 moves out of the way. And this is kind of illustrated in this morph here. You see, this is the residue which would get in contact with calcium. You see here, it engages now with calcium interaction, but once you don't have calcium, it moves away. And then it kind of rotates because it relaxes the pi helix. The rotation is around a conserved glycine hinge. You know, a glycine doesn't have a side chain. So this is often a hinge residue in proteins. Okay. Now, calcium itself is placed in close proximity to the ion conduction pathway. And calcium, you know, you have 2 calcium ion bound. These were 4 positive charges. And this means calcium plays a very important part of shaping the electrostatics of this protein. And this is illustrated in this continuum electrostatic calculation. So we are mapping the electrostatic potential along this putative trajectory indicated in orange. You see in the calcium-bound form, the electrostatics is positive throughout the pore, particularly here and then also at the calcium binding site, indicated by this asterisk. In the absence of calcium, we are missing 4 positive charges in this region, so the electrostatics gets negative. We are moving from the outside to the inside. And we would expect that, and this is a very unique property of this protein, we would expect that the calcium, the ligand itself, which activates the pore, also influences the conduction of the anions, anion permeation. And this is something we wanted to study, but the problem is we cannot study the conduction in absence of calcium because the open probability of the channel is very low. So we tried to make a trick. So what we did is we mutated this glycine. This is the hinge for the rearrangement of the helix to a proline. And when we mutated it to a proline, we found on the one hand a left shift of the EC50. So we need less calcium to activate the protein because the activated state gets stabilized. And we see basal current. And the basal current doesn't look very big, but this is probably not kind of a consequence of the low conduction, of the low open probability. It's a consequence of the low conduction. And this is illustrated here because, if you don't have the calcium here, the inside will be negatively charged, and this would repel the chloride. And this is reflected here in this strong outward rectification of the currents in the absence of calcium. And this is not, this is not about gating. This is just purely a property of ion conduction, and we can fit this to a barrier model. And what we see here is kind of, if we fit it to 3 barriers, so this would be first maybe the entrance into the neck, diffusion in the neck, and the exit from the neck from the intercellular side, then in this kind of, in this kind of current voltage relationship, we would have a large barrier here. If we start to titrate the calcium, then calcium binds, the electrostatic gets positive, and suddenly we have a linear current voltage relationship. And this is because the barrier here has decreased. So we have an electrostatic barrier which controls the excess of the anion. So in a way, you could think about an electrostatic gate, but it's not the only gate which we see here. So there is also kind of, the calcium binding does not only have an electrostatic effect, it also has an effect on conformational rearrangements. And we were interested in studying this. And, traditionally you would study this by single channel kinetic measurements, but the single channel conductance is so low. So a postdoc in my lab, Andy Lam, made a very nice study where he analyzed then the current fluctuation. So using stationary noise analysis, he analyzed the current fluctuation at different calcium concentrations, and you fit it here. It's the power spectrum. You fit them to Lorentzian functions, and you get kinetic information from them. And he had to use kind of 3 Lorentzian functions to fit this. So this means that we have probably 3 transitions. So we have modeled this here by 4, conformational states of the protein. And if we couple the 4 conformational states then with 2 calcium binding steps, then we end up with the model here. And what I just want to show you is, so what we think is that we have here a conformation where the alpha helix 6 is loose, then we get binding of calcium. At physiological calcium, we then think the helix closes, and then we get the binding of the second calcium. And then we kind of, the channel then, which is bound with calcium, transits into an open conformation, but via an intermediate, which we call a pre-open conformation. So the question is, what are the structures represented which we have seen so far? We think that the TMM16A in the calcium-free form is kind of representing the state here, right? The fully closed state. And the calcium-bound form probably represents a state here. So it's a pre-open, so it's a conformation where calcium has bound, but where the channel has not fully opened. But we were interested what's happening then in opening the channel. And the first approach we tried to do is kind of using existing accessibility studies. So can we kind of map the excess of residues to the pore? But the problem was that, we always were able to have residues accessible in this intracellular in the intercellular vestibule, but never in the neck, no matter whether calcium was bound or not. This illustrates that the neck is probably very narrow. So we did a different approach. What we did is we did exhaustive, point mutagenesis. We did alanine scanning, and then we measured calcium current, calcium concentration response relationships. And our reasoning was that, if a mutation stabilizes the open state, then we would get a left shift, an increase in potency in calcium. If the mutation stabilizes the closed state, we would get a right shift, a decrease in potency of calcium. And we were expecting that, if we hit the gate, maybe which closes the channel, which stabilizes in the closed conformation, then we would expect a strong left shift. And when we did this, then, we have here the left shifted residues colored in red and the right shifted in blue. We found 3 residues which had the strongest effect. And these 3 residues were either in alpha helix 4 or in alpha helix 6, right? And remember, we have this relationship between the 2 helices, which we already saw in the scramblers, alpha helix 6, which contributes to calcium binding and couples then to alpha helix 4. And all the 3 residues are hydrophobic residues. They are isoleucines. And when we mutated them to alanine, then you get a left shift. So you get a decrease in, an increase in the potency. You need less calcium to activate, and we get basal activity. And the residues are not in contact directly, but they narrow this region, make it very narrow. And we think that in the closed state, this is so narrow that basically it's too narrow for water and ions to penetrate. And when it opens, that it widens sufficiently to kind of allow then the access. And we know that there is energetic coupling between the helices from double mutant cycle analysis. And kind of really that we think that these are the gatekeeper which grant access to the pore. So this gave us some information again in what might be the kind of the gate region which controls access to the pore, but we knew that there was more kind of happening during gating, and we wanted to understand this. On the other hand, we wanted to understand how does the protein interact with inhibitors, how does it interact with pore blockers. And so for that reason, we started then the interaction with this compound here. The compound is an inhibitor of of TMEM16A. It's called 1-PBC. It was identified by Lily Jan's group in a high throughput screen. And if you look at it, it's in aqueous solution. It's uncharged because we have 2 titrable residue, but the pK values are in a way that they are not deprotonated. So it doesn't have a net charge. And what I'm showing you are now kind of excise patch experiment, where we kind of apply the compound to the intracellular side. And what I'm telling you now sounds a bit kind of, unrealistic, but I'm very much convinced that this is what we see here. So we add the compound to the intracellular side. And what we see is a kind of a voltage-dependent block, which is consistent with the fact that the compound is charged when it blocks the channel, and it blocks the channel from the extracellular side in a binding site, which is also in the electric field. So, and this is what we think is happening. So we add it to the intracellular side, and there probably the compound is membrane permeable. So it permeates then kind of, it diffuses across the membrane in the excise patch and then blocks from the outside. And while it's blocking, the uncharged compound gets deprotonated. And this gives then rise in this kind of, voltage dependence of block, where you have a higher potency at positive voltages, where the negatively charged compound gets kind of attracted, because its binding site is in the electric field. We also studied kind of, the selectivity of this compound. And what we found is that the compound blocks TMEM16A and TMEM16B, so the 2 channels, but it does not block TMEM16F. And this is illustrated here. So we would have here, this would be the inhibition of TMEM16A, and at the same concentration, there's no effect on TMEM16F. And this was interesting. So because there must be, there must be a difference then between these two proteins. And the interesting part is, as I show you later, is the binding side of the compound is completely conserved in TMEM16A and TMEM16F. So the same residues would be there, but the compound is not binding. So where does the compound bind? So we determine the structure here of the protein in complex with 1-PBC. And we found then the compound really, as we expected from our electrophysiology experiment, blocking the pore from the outside at the position which was residing inside the membrane. And this is what you see here. So this is the cryo-EM density. So the density is actually pretty nice. So you see here, this is the surrounding residues of the protein. They're very well defined. This is kind of, given that it's such a small compound, it's kind of a pretty good cryo-EM density to fit this in here. So if we are looking at the surface, then it's obvious that the compound binds to a pocket. And the pocket approximately fits the shape of the inhibitor. And we think that this pocket is only adopted in the open conformation. So it's an open state blocker. So if we are looking then at the surrounding residues, then we see that, we have a lot of interactions. We have, the interaction map the polarity. So we have hydrophobic interactions to the hydrophobic part. But we also have here the interactions between these lysine residues and this hydroxyl residue. And we think that this interaction then shifts the pK of this hydroxyl group so that the proton dissociates and the compound becomes negatively charged. This is illustrated in this experiment when we mutate the lysine residue to an alanine. No, we couldn't mutate it to an alanine because it was not active. We mutated it to a glutamine. And what we saw is that kind of, this would be the voltage dependence at +80, -80 millivolt of wild type. You see, we see a kind of a strong decrease of the potency of the compound and a complete loss of the voltage dependence. And we have another positive charged interaction, and this is arginine 515. And this arginine 515 is a residue which kind of, what we were kind of working with, what we were wondering about this for a long time, because we knew from early experiments that if you mute this, it looks like that this is a central player in shaping the conduction of TMEM16. And you see here the residue makes here interactions, it stacks on the compound, and also its mutation has a strong effect. Decreases the potency, loses most of the voltage dependence. What we have also found is, it's something, in this data set, it's something which we were wondering a lot of a long time before, because what we knew is that alpha helix free in our original data set was never well defined. So the entire structure was good. Alpha helix free was not defined. And we realized that it was not defined only now when we determined this data set here, because it can adopt two different conformations. So which means that in the presence of the compound, this alpha helix free adopts an up conformation where then arginine 515 interacts with the blocker. In the calcium-free conformation, it adopts a down conformation where you see that first the helix here has, the arginine has rotated out of the way, and this this tyrosine is instead kind of has moved in because there's a kind of a downward movement of the helix, and this this tyrosine then blocks the binding site. So in a calcium-free form, the binding site of the blocker is not adopted. And you see here a morph of all the conformations which we have, and what you see is kind of looking a bit funny. It also looks a bit funny to me, but this is basically from the conformations we have. You see here this remodeling of alpha helix free, upon calcium binding, so in compound. So you see that it looks like that in this gating, there is a remodeling of this extracellular binding site. And then only which then kind of makes space for our compound. So in our kinetic model, what we think is that, we think that this remodeling is happening maybe from this tight to this pre-open conformation where we get the remodeling of the extracellular part. We think then probably there we form the binding site for the compound, and we think it might probably be able to block both states, the open state and the block state. And we think that the crystal the cryo-EM structure which we have might resemble this blocked conformation, because we see that kind of, if we map the pore diameters, it's still a bit too narrow. So we think the pore might widen a bit more. So this is kind of in a nutshell. So what do we think happens during gating in TMM16A? So in the absence of calcium, we have negative electrostatic on the inside. Calcium binds, the electrostatic changes, so it gets now attractive for chloride, but there's still no conduction because we have the gate which is still closed. It's only kind of upon transition from the pre-open to the open conformation that the gate moves out of the way, and then we get calcium permeate. And we think that these two conformations are blocked by one PBC. So at the very end, and I kind of realized that we are running a bit late, I would like to just tell you a bit more recent experiment on the last kind of clade of the family of TMM16 protein. It's TMM16F, and TMM16F is very interesting because it shares properties of both kind of branches. It's kind of it has properties of an ion channel, and it has properties of a lipid scrambling. Now I should mention that it might be that there is also conduction which is conferred by NH-TMM16, and there's work by Alessio and other people who think that they might be conducting. But TMM16F is in a way a very well-behaved channel, and it has channel features which are really kind of reminiscent to classic channels, and this is seen here. So we are looking here at at conduction, at an excise patch current of TMM16F at positive and negative voltage in saturating calcium concentration. And there are some features which are different from TMM16A. So first, the currents are not selective, or even if they are, they're even slightly cation selective. The other feature which is different from TMM16A is that in saturating calcium at negative voltages, you get this time-dependent closing. So you have really voltage dependence, etc., in calcium here, and you see this relaxations here. So this means that there are properties in TMM16F which are probably different from TMM16A. The same protein kind of also scrambles, and we have done this by reconstituting the protein and then kind of starting scrambling at kind of 0 calcium. We don't see scrambling. At high calcium, we see maximum scrambling, and at intermediate calcium, we see then kind of an activation of scrambling. If we kind of look at the concentration response relationships, then you see that we get in both cases activation, and the activation, keep in mind, these are two very different experiments. I think it's remarkable that we think that the calcium potency is about similar. So we think that both processes are kind of activated by the same calcium step. We then started to determine the structures of TMM16F. And, in the presence of calcium and in the absence of calcium, and I remember very well because, the first structures which we were seeing here was while I was at 2017 in Mexico City at the Ion Channel conference, and we were kind of puzzled because the structure looked very much like TMM16. So it didn't have the feature we expected the scramblers would have, right? And this is just the morph between the calcium-free and the calcium-bound conformation. You see that there's a rearrangement of helix 6. It's not as extreme as in TMM16A because we don't have this other helix, we don't have this pi helix formation, but there is no indication of an opening between alpha helix 6 and 4. And I just want to kind of show you a couple of experiments which are not yet published, but where we wanted to kind of understand better how is TMM16F activated. And if we assume that we might have a similar kinetic scheme as in TMM16A, then we think that the structures which we have seen here are also kind of reflecting the calcium-free closed form and maybe the calcium-bound closed form, right? And what we think is that probably kind of gating would would proceed in a similar mechanism, because it's at the end it's very similar to TMM16F A, and we thought at that time eventually maybe what makes the difference that in the open conformation maybe you have an equilibrium between two conformations, one being an ion conductive conformation and the other one maybe a lipid conducting conformations, and maybe they're in equilibrium. Actually, I was very fond of this theory and I still like it, but I don't think it's true. Okay, and the reason why I don't think it's true is what I want to show you now. So we wanted to study the activation of TMM16F, and so we did similar experiments which we did for TMM16A. So we did extensive mutagenesis of the pore region, and then we measured two quantities. We measured here the calcium concentration response relationships of the currents, which we plot here on the x-axis, and we measured in a cellular assay lipid scrambling. So there we measure the binding of annexin to the surface of cells. And, we found that there are mutants, we did this either a basal scrambling at resting calcium concentration or at higher calcium, but we found some mutants which are already highly active at, at basal calcium concentrations. And these mutants are all located here in the upper right corner. So they are have a left shift in the calcium response relationships for the currents, and they have large basal scrambling. And the three positions I want to point you to are here, these three positions, and they are mapping here at alpha helix 4. This is the helix we would expect to be involved in the opening again, and also two positions on alpha helix 5. So which is supposed to be the core of the subunit cavity in NH-TMM16. And the same positions have previously been also identified by Wang-He Yang's lab, and he tried based on initial computer simulations, and then also tried by mutagenesis. So we then tried to look at this position and look at, here, calcium response, current response relationships here at the polar nature. And what we found is the more polar you make the residue, the stronger the left shift is. And the most dramatic conformation is TMEMS, if we mutate the phenylalanine 518 on alpha helix 4 to a histidine. And we did the same experiment in taking the mutant, reconstitute it, and look at the calcium dependence of scrambling in vitro. And we find here the wild type, and we find the mutant has also a very strong left shift. So it looks like that this mutation stabilized the open state of the scramblase and of the channel. And then we started to determine structures. And we determined structures in the absence of calcium and presence of calcium. And what you see here, so we compare the structures now to the structure of wild type in the presence of calcium. And this is always shown here in this transparent black. Okay, so in the absence of calcium, the conformation of this mutant F518A is exactly the same as we find it for wild type. So there is no difference. But once we add calcium, have a look what's happening. We determined two structures, one in nanodiscs and one in detergent. We are looking in comparison to TMM16F wild type in calcium. You see, in the presence of calcium in the nanodiscs, we get a closing of alpha helix 6 because it engages in calcium binding. But then we get a conformational change of alpha helix 4 and alpha helix 3. And this conformational change is even more extreme if we look in detergent, right? So, and what we have is two movements. We get a straightening of alpha helix 4, which is bent in the closed conformation in wild type, and we get a bend here around a conserved glycine hinge, and also a straightening, and also a conformational change of alpha helix 3. And this causes a distortion in the membrane in a way as we have seen it in NH-TMM16, but we have not seen it in wild type TMM16. And this is illustrated here. This is the nanodisc structure. This would be then the region of, this would be here alpha helix 4. And you see that this kind of, the density is getting weaker. And this is now a morph of all the conformations of TMM16F, which we have. And it looks wild. I mean, we are going from a calcium-free conformation right here. And then we bind calcium, and then there's a different transition. And maybe have a look here at the relationship of these three spheres. So what's happening is alpha helix 4 rearranges, and there is kind of the relationship between these helices is changing. Because what we see is not a complete opening of alpha helix 4 the way we see it in NH-TMM16. We see still kind of some residual contacts, but the contacts are much smaller. And there is a rearrangement of these helices, and we have tested this by also by double Newton cycles. We see that these residues, which are far away in the calcium-free state, are actually coupled energetically. And what we get is kind of, a rearranged structure which has something like a pore. And this is something which you see here when you look from the outside. This is kind of a pore of the calcium occupied mutant. And this is kind of just where you look at the changes of the pore diameter from here. The blue would be the no calcium, and we're looking from the outside to the inside. And you see this expansion of the pore diameter in the presence of calcium. So there's a big expansion. And what you see here is kind of, the region now, we are looking here from the top, we are looking from in the membrane. So we get basically here the pore. And what you see once it comes here, so this would be the outside of the protein. So we have these small regions where the two helices are in contact, where the cavity is not fully open, but this is not a very long distance. So this is only about, let's say, maybe half of the distance of the hydrophobic core, or even less. So what we think is the following. So we think that we have one conformation which eventually might be able to catalyze two both processes. And that the catalysis of both processes might be in slight, at least part of it be on the same pathway, like here, we get the rearrangement, the rearrangement exposes then hydrophilic residues to the outside. This distorts the membrane. So this distorts the lipid bilayer. But then we have this region here where the pore is kind of, closed, where probably the ion conduction pathway is separate from the lipid conduction pathway. But we think that maybe with these conformations, we can still kind of fulfill both kind of properties, because we tested a couple of other mutations to see, would we see any structures where we get a further opening of the cavity, but we have never seen it. Okay, so with this, I'm coming to my last slide. And just to summarize what I wanted to tell you today, so we have here one structural unit, which we call the subunit cavity, or the pore in TMM16A. And this kind of allows then, it shares very similar properties in general in how, this unit is activated by calcium by binding to a conserved site, but we also see features which are unique and different. In NH-TMM16, the binding of calcium causes kind of this opening of the subunit cavity. So alpha helix 4 gets out of contact with alpha helix 6, opens a cavity, which provides a pathway for lipid head groups to cross the membrane. In TMM16A, an initial idea was that maybe kind of, there might be a pore which would have been lined by lipid head group. And this, of course, was a very intriguing idea. But in our structures, it looks like really the evidence is that this TMM16A is really not opening up on its entire way to the membrane. So we instead have a water-filled pore. Of course, there might be some lipid contact on the intercell. TMM16F, our initial idea was, since it has both properties of an ion channel and the lipid scramblers, our initial idea was that we might have a kind of, an equilibrium between the two conformations. So one looking like in NH-TMM16, one is in TMM16A. I think with our new data, we are more thinking like this, that we might have one conformation, and that this one conformation promotes then both kind of catalytic paths, and that's in some parts these ion lipid conduction would happen in the same region, but in the center they would be basically happening on distinct trajectories. So now that's my really last slide, and this was about 13 years of work, so there were many people involved, very very talented people. The first experiments on NHT-MEM16 crystallography was done by Janine Brunner, who really did a marvelous job there. Novandi Lim, who also contributed a lot to this. The TMEM16A work was kind of initiated by Cristina Paulino. He was the first person in my lab starting with cryoelectro microscopy. And then all the electrophysiology and the later structural work also with the inhibitor and also the analysis, by the kinetic analysis, was done by Andy Lam, a really very good electrophysiologist. Valeria, in collaboration and with Christina, when Christina moved to her lab to the University of Groningen, has done the work on the activation of NHT-MEM16. And Carolina, then also initially with Christina, has worked on, and together with Novandi, has worked on TMEM16F. And the last work I've shown you was work of Melanie and Carolina on the activation of TMEM16. And I wanted to thank Kwang-Hee Yang for providing us his TMEM16F knockout line, which was very important for the screening process. I showed you. And then our crystallization facility for the beginning, we did a lot of crystallography, but then, and also the beam lines where we collected the data. But then later, all the structures were done by cryoelectro microscopy at our institution. This is the funding. And then I thank you for your attention, and I apologize for being a bit long. I think you're muted, Jorge. Jorge, your microphone is off.
We have about 5 questions.
Sure.
I will read one to you, one by one. So Elena is asking, does calcium triggers similar conformational changes in scramblases that function through the out-of-group mechanism?
Not exactly the same, right? So if you think, if you think about the if we say that TMEM16F works by the out of groove mechanism. Okay, maybe I have to be more precise. So, because, in NHT-MEM16, we have the opening of the subunit cavity, right? And we think that this opening of this polar cavity provides a pathway for lipids to go across the membrane. But it might well be, and I think data from his recent data from Alessio Arcadi's lab shows that, lipid movement could also, not all of the lipids necessarily have to, move in the contact with the protein. So maybe the destabilization is kind of sufficient that you might also have lipid flip-flop without having a direct contact, right? And I think this is plausible to some degree. So I think this is really, at the end, you're kind of lowering energies, and at some point, the question is, what is the transition energy? But I think Alessio has kind of good evidence that there might be something like this going on. And this means that you could have out-of-the-groove scrambling also in NHT-MEM16. But if you kind of now compare TMEM16F and NHT-MEM16, I think then the movements are a little bit different. So in NHT-MEM16, we have something like this clamshell opening, right? So it's like really kind of a lateral opening. While in TMEM16F, we have a rearrangement, a dissociation of alpha helix 4, and then maybe a reformation of contacts on the intracellular side, where we don't have this entire cavity being open. This is honestly for us, it was initially also kind of a bit difficult to take. So do we think now that this might scramble this conformation? Right? It took us a while to accept this also, that despite the fact that the cavity is not entirely open. But we think, yes, it might be. And then this would also be support that there might be a bit of an out of the groove scrambling, because, for some distance, at least the lipids would have to kind of cross the outside of the protein.
Okay, the next question is, why do you think there has been so challenging to obtain an open pore conformation structure for TMEM16A?
It's a good question. It's a very good question. And the answer is, I don't quite know. You know, I think it's not, you know, in a way, we were so lucky when we determined the NH-TMM16 structure, because it has told us, you know, we didn't know anything about how such a protein works, and it has told us nearly everything. And this is kind of, you know, I think kind of having conformational preference is not, you know, if you think about the Boltzmann distribution, right? And then check about, you know, let's say, when do you see a conformation? Let's define, we see a conformation if we see 90% in this conformation, right? Then calculate how much must the energy difference be that you have a 90 to 10% population difference. And then maybe the energy difference is really not so big, right? And for some reason there must be, you know, for some reason there must be something which stabilizes then one conformation or the other. And it's a good question, because, you know, we are trying a lot, and we did try a lot to see kind of, you know, would we see more expansion, you know, in studying mutant, you know, we studied some mutants which really shift then the concentration response relationships very much to the left. And we still, you know, the question is, we also don't quite know how big do we expect it to be, because we know that the pore, or we think the pore is narrow, because all our accessibility studies kind of indicate it's narrow. I think your studies on where you studied, you know, the effect of isothiocyanate in stabilizing the pore, I think is, in my opinion, also most compatible, that there is not a very kind of wide pore.
So what do you think about PIP2?
So
and its role in the widening the pore?
You know, I think a lot what other people probably also think, because, you know, based on data from others. So I think it's reasonable to assume that PIP2 stabilizes an open conformation.
Okay.
And you know, seeing all these conformational changes of the helices, including alpha helix 3, to me, it would be plausible that, you know, that maybe PIP2 would stabilize a conformation of alpha helix 3 on the backside.
Mm-hmm.
We have never really been able to localize this. We often kind of use PIP2 in our preparations, but we have never seen it. So, but you know, this looks, I think, kind of, you know, given that there is so much kind of conformational changes there, I would find it plausible. But, you know, any other position is also fine for me.
Okay. So
but I think it's not unusual, kind of, you know, it's not unusual that you see conformational preference. Yeah. And maybe just one quick remark: we found something very interesting in some of the TMM16F activating mutants, which I have not talked about. But there we found kind of, you know, we found two populations. One population where we find both subunits in a closed conformation, and another population where we find subunits, one subunit in a closed and one in an open conformation. Mm-hmm. And initially I was kind of a bit confused, but this is what you expect, right? Because if you have, let's say, an open probability of 20 or 30%, then the majority, and if you have independence between the two subunits, then the majority will be kind of, you know, close. And you will not see any open, but you will see then closed open. And this is something we have seen a couple of times with TMM16F. So I think kind of by tuning the energetics, you will be able to shift between conformations.
I see. Okay.
And you know, in occasionally, I mean, of course, it's a dream when you start to see all the conformations in one data set, as we have seen in NH-TMM16, right? In the calcium-bound data set, we saw different conformation, and it probably just reflects that they are energetically similar.
Yeah. Okay. So the next question is, in the results you presented, it is clear that one PVC doesn't block the current of TMM16F. Do you evaluate if the drug could inhibit its scrambling function?
So we did not do that yet, but we will do that, and I would guess no.
Okay.
Because, you know, what we think currently is, and I think it's a bit controversial, we think that the pore, the active conformation of TMEM16F is looking different to TMEM16A. And if you see in TMEM16A, in order to bind one PBC, you really you have a pocket which really is shaped to really be nearly complementary. So if we have the opening as we see it in TMEM16F, we get a widening in the regions where there would be where the one PBC pocket. And then I wouldn't expect that you would be forming this interaction.
Okay.
But but it's a very good question, because it would be nice if we would be able to see a blocker where we could block the ion conduction pathway, but not the scrambling, right? Mm-hmm. And I think there must be something like this.
Okay. Eduardo is asking, in a calcium channel, what made it specific to calcium and not to other ion? I suppose it's a divalent cation.
In the calcium channels.
In a calcium channel, but probably he's asking in a calcium activated chloride channel.
So if you, if you're talking, usually, okay, calcium has a very high, has a very large net charge. So, and you see that you kind of, in the binding side, you have to provide a lot of negative charges to compensate for that. So I think binding calcium really requires a good coordination in the way how we have seen it in our binding site. And actually, if you compare this binding site to similar sites in the calcium pump, the calcium ATPase, then you see similar features. The interesting thing of the selectivity of this site is that you can activate the protein with calcium, you can activate it with larger ions like strontium, you can activate it even with barium, but you cannot activate it with magnesium. And this is something also because we have also been studying recently a magnesium transporter. It looks like that the problem with magnesium, it's even smaller than calcium. So you need more energy to dehydrate magnesium and coordinate it well. Mm. And this is why magnesium is often bound with still some of its water around. And then the binding site has to look different. So this is probably the reason why it does not bind magnesium there. And for, and similar things are for the channels, right? In calcium channels, you often have a higher field strength to be able to interact with the ion.
Okay, next question is, what would be the evolutionary advantage of having both a pathway for ions and one for lipids?
It's a very good question, and I don't have an idea. I don't have a clue. I think we don't understand currently what is the physiological function of the conduction of TMEM16. It would be, I would really want to know this. I mean, there was a theory by Lilian when they found it. She was characterizing, she found the conduction of TMEM16F, because this was at the time when it wasn't clear, is TMEM16F itself a real scrambler? And there she found that it's cation selective, and she also found that eventually it might also permeate calcium. And the idea is that it might have a positive feedback, so it could diffuse its own calcium. I mean, another idea by somebody, you could think about if you transport a negatively charged lipid, maybe it allows you to transport the counter charge. Maybe, could, but I really don't know. It's really interesting. I mean, the other interesting thing evolutionary to think about, and I don't have an answer for that, is probably in evolution, I would guess that the fungal scramblases are the first one to pop up.
Yeah. Okay.
The fungal scramblases are actually the one which are an intracellular, on the intracellular side, probably in the ER or something like this. The question is, why are they calcium dependent? And why are they not strongly calcium dependent? Because, the strong calcium dependence is, TMEM16F has a very strong calcium dependence. It does not scramble in the absence of calcium. while NH-TMM16 scrambles, right?
Mhm.
The question is, if this is older, why has the calcium binding site evolved there and only became kind of more sophisticated or better regulated than in the later stage? Something which I find fascinating, but I don't have a good answer.
Okay, I have a couple questions for you. One is, what's going on with TMM16B? Why is is a channel, but nobody else knows about the structure, and seems to me that is relegated to a side. Why is that?
I don't know. The question is, I don't know why not more people are studying it, because it's actually very interesting. Mm-hmm. I know biochemically it's not very stable. So we are in a way kind of, as many of the other ones are. So in a way, we are kind of lucky that TMM16A and TMM16F are reasonably stable to work with. So TMM16B is in its stability less well behaved. I think it has many properties very similar to TMM16A. I think also the fact that it's kind of blocked, the blocking mechanism, that the blocker binds there is also kind of a bit relieving. That say, well, it cannot be so different.
Mm-hmm. Yeah. Okay.
But on the physiology, this is of course a completely different story. This, I'm aware that whatever I've told you, this is biophysics. Of course, the important thing is where do these proteins really participate?
Mm-hmm. Well, the last question is, so this membrane deformation that is produced by the movement of the helixes, do you think that the channel is sensitive to force?
It's a good question. It's maybe a question I would rather want to have an answer than have an answer. I could imagine. I don't know how, I don't know how compressible the membrane is. I don't know whether somebody has studied it. Are you aware of some indication?
No. I just stumbled with the Roderick McKinnon last two papers in PNA and PNAS, where he showed that you can calculate the effect of the force on the channel, on piezos, just by analyzing the effects on the what is this, vesicles. So I was wondering if you can do.
I would expect, I could imagine that there is some degree of. And I'm just wondering. So Alessio has kind of in his recent work, he looked at kind of reconstitution of NHT of I think he used a different fungal homologue, but reconstituted it in different lipids, and then found that the conformational properties of the protein changed. So that he found different distributions of kind of open or closed conformations.
I see. Okay.
So I think in theory this must be, but I don't know how strong it will be.
Yeah. Okay, I understand. Okay, I guess we don't have more questions. Elena, thank you. So everybody else, thank you very much for the very interesting talk. I appreciate that you accepted our invitation. Thank you very much, Raimund.
Yeah, thank you.
I don't know if Eduardo